Hello everyone, hope you're all doing well and healthy. So this video today is to talk about the data analysis of the data we collected in our labs. But first I'm gonna go through a few announcements. First, about forest soils, thank you for watching all the presentations, writing comments, having a bit of a discussion with the speakers, and submitting your assignments. They've been graded, and you can see your grades online. For those that submitted late, we knocked off 5% per day as per agreement. And but in general, you all did super well on your answers. If you left any comments for the speakers on Panopto, go ahead and go back there. They responded to your comments. About the midterm, the deadline, don't forget, it's on the 12th at 11:59. I see some people have already submitted. For those that are still working on it, please continue. If you have difficulties submitting on Blackboard, email me and we try to find a better arrangement. As I said on the announcement, you can write on a piece of paper and send me the photos. Anything, we're flexible at this time. If you have any questions, let me know. Okay, and on the survey I sent last week, I'm very glad I did that survey because I could identify many of you expressed that the presentation we had planned for this course would be a bit too much to handle nowadays, given that we're all working remote. And I imagine coordinating with 5 other team members must be a bit challenging nowadays. So I think it's best if we maintain the data analysis aspect of it, because it's a skill you will learn, a little bit of Excel graphing as well. It's a skill you're gonna learn and use later at work or in other courses. And we still get to see the results of your lab experiments. So we're gonna go ahead and prepare a lab report, and you will submit your lab report individually, not as a group. You can work as a group if you want. Within your group, you can all have the same answers if you want, but you have to submit individually, okay? And the report can be about 1 page, maximum 2 pages. You can increase the number of pages by adding your photos, but the purpose is to have something simple that you can do. It won't take too much of your time to prepare, but will reach the purpose, which is for us to calculate your data, look at it, and make the interpretation. That's the purpose of this lab report. Okay, the deadline will be on the 24th, so we still have a few weeks. And on the next slide, I'm going to go over the components that we need to see on your lab report. Okay, and so after we talk about the components of the lab report, we're going to go over the calculations for your data. First, we're going to talk about the calculations for soil respiration and then for enzyme activity. So today's video has the purpose of going over these 3 things: the lab report, the data for soil respiration, and the data for enzyme activity. So going over the lab report. The lab report has to have these 4 components. The first component is the description of the things you're comparing. So some people are comparing samples that were collected from near the trees or from different types of trees. You know what your group decided to compare. I think most people are comparing distance from the rhizosphere of the tree, so collecting samples that were at the rhizosphere, and the other set of samples were collected like 10 meters away or something. So you need to describe these samples, okay? You need also to mention the number of replicates you collected. Most of you collected 3 replicates per location. Mention also where you collected the samples. Was it in McAllen? Was it in San Benito? Where was it here in the RGV? Okay. And also make sure to describe what type of amendment you used. Some people used motor oil, some people used Gatorade, cigarette butts, bunch of things. Mention what amendment it was. Okay, so that's the first component of the lab report. It's the description of your samples. And the second component, it's about the objectives. So here I want you to write why did you compare the samples? Why did you choose this type of tree versus the other, or this distance from the tree versus the other? And then touch a little bit on what you expected from this comparison. More biological activity here, more biological activity there, and why? Okay? Also, why did you choose these amendments, like your Gatorade, for instance? What did you expect to see from it? And is it common to see this amendment being added to the soil? Maybe it's part of an environmental problem. Maybe it's part of a management to make the soil better. So maybe you write a little bit about this under your objectives. Okay. Then the third component you need to have is your results. On your results, I want to see 1 bar graph for soil respiration, showing the data of soil respiration across all your samples. And the other bar graph I want to see is about this enzyme activity. And within the results, I also want to see some interpretation of the graphs. So for each of the graphs, you're going to write 2 or 3 sentences saying what you're looking at the graphs. Okay, so all the amendments increased soil respiration, and samples taken away from the tree had lower soil respiration than in the rhizosphere. You're gonna have to look at the graphs and describe what you're seeing. Okay, then the fourth component of your lab report, it's your conclusions. So make sure to restate the main findings. For instance, your main find could be that amendments increased soil respiration or increased enzyme activities. And also say whether or not you obtained the expected results that you described on your objectives. And also write what is the importance of your amendments to the environment. Okay, by adding Gatorade, did that suppress the bacterial activity? Or by adding banana peels, increased microbial activity? Something like that. Write about the relevance of the data, your environmental relevance of the data you collected. Let me write here, environmental relevance. Also, another thing I didn't mention, it would be nice if you compare the data you obtain under soil respiration versus the data from the enzyme activity. Did both give you the same type of results, or it wasn't the same trend across all your samples? For instance, if you added Gatorade increased soil respiration, did you observe the same in enzyme activity? Make a note about that on your conclusion as well. And don't forget to add all those awesome photos you took, okay? Okay, now everyone, so moving over to soil respiration. In class, you calculated how much soil respiration you obtained from your samples using this equation. If you don't have this number, someone in your group must have, because we did the calculations in the classroom. So pretty much you have the volume of acid you spent to neutralize your standard and the volume of acid you have to neutralize your sample. Then you multiply the difference by the molarity of the hydrochloric acid, which was 0.5 molar, here, 0.5 molar, times 6 times 1,000, those are correcting factors, and divide by the sample weight. The sample weight was 50 grams. That was the amount of soil we used at the beginning of the incubation, 50 grams. And we incubated for the period of a week. So you have to calculate how many hours you have in 1 week and use this number here in the equation. So by plugging in all the numbers into this equation, you will obtain the amount of CO2 in milligrams of carbon dioxide carbon that the microbes released for 1 kilogram of soil for the period of 1 hour. Okay, because what we're doing here is estimating soil respiration rates. Okay, how fast were the microbes releasing CO2 within those jars? We could have calculated this per gram of soil for the 50 grams of soil for the 1 week, but it's more common to find in the literature this data expressed per kilogram of soil, so we're extrapolating for 1 kilogram of soil. That's why we're multiplying here by 1,000, and we also express it within the period of 1 hour versus the period of 1 week. Okay, so you should have this number, your group should have the number for each of your samples. So pretend I have a group and we did these incubations for samples that we collected in the rhizosphere of a tree and samples that were collected distant from the tree. So at the end, I can have a table that looks just like this one. So on the first part of the labs, we incubated the samples from the rhizosphere and from the distance from the trees. And then I calculated my soil respiration rates, and this was the numbers I obtained. Then on the following week, my group and I, we added an amendment. Let's say we added charcoal. So we added charcoal to all our samples, to the same samples we had before, the rhizosphere soil and the distance from the tree source. And this was the number of the respiration values we obtained. And in this case, I want to make sure that you notice that here we have 2 replicates per treatment. We tend to call this a treatment. Rhizosphere, distance from tree, the charcoal, okay? We can call it a treatment combination or a combination or a simple set So within each sample set, we have two replicates. Okay, so replication 1, replication 2, replication 1, and replication 2, and so on. So I'm gonna input this table into the Excel, and we're gonna start doing our graphs, okay? All right, so moving over to the Excel part, I exported this table with my fake data into Excel. So you see all the samples, all the locations, whether it had an amendment or not. In this case here where I'm highlighting, it didn't, are the results of the samples that did not have any amendment. And so we have the samples collected from the rhizosphere, collected samples from away from the tree, and you'll notice that I also list here the replicates. And on the second set, I have also samples from the rhizosphere, from distance from the tree, and in this case amended with charcoal. And here I show the respiration rate that I calculated using the formula I showed before. And you see that the values vary depending on the type of samples. It's also expressed in, as I discussed before, milligrams of carbon per kilogram of soil per hour. So what I want to get done with this Excel exercise is to first make the average of the replicates for each sample set, and then calculate the standard deviation of the average. The standard deviation is how much the samples vary from the mean, how much it's the dispersion of data from the mean, okay? When we look into a population, we may have a dispersion of heights, as I discussed with some groups in class. We may have an average height for the valley, but we have much taller people than the average, and much shorter people from the average, okay? We're making now here the average for the sample set, and so I'm gonna average out the soil respiration for each of the sample sets. So you see how I enter a automatic function of Excel called average. So equal to start the function, equal sign to start the function, and then average, and then I select the range of samples I want to average out. Now I'm going to calculate the standard deviation, which is the dispersion from the mean values. Okay. So there's also an automatic function. It's called stdev, which calculates automatically the standard deviation from our sample values. And we're going to do it for each of our sample sets, looking into the dispersion of the replicate data from the mean. I hope I'm not overcomplicating things, okay? Just slow down the video and follow the Excel exercise. So now we're going to do a bar graph. And to do a bar graph, I like to have the data organized in the sequence that I want to see my bar graph. So I'm going to copy again the name of the samples and their average values and the standard deviation. So you see, so the first thing I want to show on my bar graph is the rhizosphere data without any amendment. I'm doing voiceover of this video because it's easier to just follow along versus try to explain what I'm doing while I'm doing. So you see, I'm organizing here another table where from here I'm gonna directly create the chart. So first we're gonna equal sign and copy that value from there. So just the equal and select the value you wanna copy. Equal, select, equal, select. Okay, let's copy now the standard deviation values. Equal, select, equal, select, equal, select. Note that I'm here building this new table in a different order from my original table, right? So that's why we need to be careful here. Okay, so now we have the table with the means and the standard deviation for each sample set, and I'm going to select this data and create the chart. So you go on Insert and choose your bar graph, and voila, here is the bar graph for that selected data, and I can change the chart title and add respiration rates. All right, so we write a title for the plot, for the chart, and now we're gonna add the error bars, the standard deviation bars to our graph. So we go under Add Chart Elements, all the way to Error Bars, More Error Bar Options, and then we go under Custom. And then you customize it by selecting your error bar values. First you add the positive values, which go on the top of the bar, and then you choose the negative values, which goes under the mean values. Okay, then you OK, and here is our error bars there. My data didn't vary that much, so my error bars are not that big. You may find bigger error bars on your average values. So we made the average for the replicates. We calculated a standard deviation of the average. We made the bar graph. We inserted the standard deviation bars, which we call the error bars. Now let's add labels to the axis. So let's go again under Add Chart Options and add a element to it, the title to the y-axis. So I'm typing respiration rates and the units, which we're going to express as milligrams of carbon per kilogram of soil per hour. And we have our graph. So the graph is now ready to put on the report. Let me know if you have questions here, and I'm happy to go over with you, or you can slow down and watch it again. Okay? Right. Now about the enzyme activity using FDA hydrolysis. FDA stands for the fluorescein diacetate. So we chose this method, the fluorescein diacetate, because when enzymes in the soil are working, when the microorganisms are active in the soil, releasing a lot of enzymes to break down organic materials or to scavenge for phosphorus, when the microbes are active, they are able to hydrolyze, to break down this fluorescein diacetate that we're adding to our samples. And then in the laboratory, we used the samples each group collected. We added the reagents, substrate, we made a standard curve. So we're going to calculate how active the microbes are based on the speed they break down fluorescein diacetate. All right, and the more active they are, the brighter the color they're releasing in those tiny little wells we're working. Okay, so you're gonna have to bear with me because it's going to be a long process to go through these calculations, and it's the first time I teach how to do these calculations without anyone's feedback to know whether everybody's following or not. But I'm glad I'm happy to answer any questions over email. So this is our protocol. And so you want to go over to see what we added and such. But most importantly, this is the map of the samples you all had. And this is our plate, the layout of our plate that we had in the lab. And in this first two rows, we added the standard, which are our reference samples. And then here is our actual samples. And then here we added the blanks. When I say here, I'm referring to each of these wells. So on A1, we had no standard added. On A4, we had 10 micrograms of the fluorescein standard added, and then we increased to 20, and then we increased to 30, and so on. Then we added our samples, and then we added a blank, and then we put it on the instrument, and we made a reading. And this was the document we got from the instrument. Okay, so this is an example of data I'm using just to go over this exercise with you. So here you have a few information about the conditions of the running, such as when it was run, what time, and what type of plate we used, what was the wavelength that we used to measure the color of the samples. So this is probably group 2 data. I remember they had a very nice standard curve, so I'm using their data, and each group knows how they distributed their samples on their plate. And group 2 used this layout. If your group did a different layout, you should have taken notes, okay? You should have written documentation on this. So this is groups 2 data. They're gonna have to do their own calculations too. So first I want to do the standard curve. What is the standard curve? So what I get here is absorbance data, correct? And you see in any material, any solution has an absorbance to light, correct? So in the case of our samples, they were yellow, and the brighter this yellow, the more light they would absorb from the instrument. So here all I have is the numbers of absorbance. That means nothing in terms of microbial activity. But I do have reference samples, which I call the standards, and they mean something. I know how bright they need to be in order to indicate microbial activity. So first I'm going to make a curve, and based on this curve I can calculate what was the activities of the microbes in our samples, okay? So first we type here the concentration of fluorescein, which is our standard, and here we're gonna average the absorbance, okay? So if we're following the standard layout of the plate, at every 3 samples on the plate should be 1 standard point, correct? So let me see if I can keep both visible. If I'm going too fast, there is an option that you slow down the speed in which the video is being shown and the voice and such, okay? Okay, so let's average. Oh, This is, my screen is too small. Okay, but it does help a little bit. So first, I'm gonna average the absorbance on my standard that received no concentration of fluorescein. Okay, so average, and then I select the three together, and then here average for the standards that receive 10 micrograms of fluorescein, which is in this range, and then the next one should be 20, which should be in row 7, 8, and 9, and average 10, 11, 12. 40 should be in 13, 14, and 15. 60 should be in 16, 17, 18. Is it correct? 60, 16, 17, 18. Okay. 80. 19, 21. Okay, so this is just to average out the absorbance across your standard curve. Okay, and I'm using this group's data because during the activity I looked at their data and it was well, let me just double check here. 200, 207, yes. And during the lab I looked at their data and they did have a pretty good standard curve. They didn't have many pipetting mistakes, and their standard curve was quite linear. So that should be less. Very well. Okay, so here we have our the average of the absorbance across each of these standards. Now we need to see the standard curve. What is the trend that this data is distributed? So we selected the data, we selected everything, and then we go under insert, and then we ask for scattered plot. Okay, so you see as we in the x-axis we have the concentration of the standards, and on the y-axis we have the absorbance. Okay, and you can see it's quite linear. As we increase the concentration of the standard, we increase the absorbance. We call this a linear relationship. And the more straight this line is, the more linear the data is. So I like to add a straight line here. So I selected all these points and right click and ask for add a trend line. And I ask for the linear. Okay. Then I also wanted to see the R squared. So the R squared indicates how along this trend line the points are located. If it deviates from this straight line, the R squared will start to decrease. So you see, it has an R square of 0.98. I like R squared of 0.9099. But it's the first time you guys did this, so we should just learn with this experiment. So you see, this point, it's deviating too much from the trend line. If it was closer to the trend line, the line would have been more straight, and the points would have been more straight distributed, and the R square would be 0.99. So this is our correlation coefficient, okay? I also would like to see the equation. So the equation helps me to, you see this equation, y equals 0.0033x plus 0.0745. Okay, so this equation has a very important purpose, very important. We're going to use this equation to estimate the concentration of fluorescein in our sample. Because for our samples, all we know is absorbance. We need to translate absorbance into the concentration, okay? So we have this equation, we're going to leave it here and use it soon. Do you guys want to add the axis to this chart? Let's do it. Add axis. So the first one we're gonna, axis titles. Let's add to the vertical axis. So here it's absorbance. I'll add also a title to my horizontal, which is concentration of fluorescein in micrograms. I'm writing micrograms here, but usually we use the symbol for micro or just a UG. That means micro. And let's change this. Let's call it standard curve. Very well. So we do have the standard curve. And if you guys remember, when you're studying either chemistry or math, you have this equation. The y represents the values in the y-axis absorbance, okay? The 0.033 represents the slope of this curve, all right? The slope of this linear relationship. It's the angle of inclination. The x represents the concentration in the x-axis. And 0.0745 represents the intercept where the line crosses the y-axis, okay? So I can, Excel can calculate for me this slope and the intersect. I can ask, let's ask Excel to do that for us. So first we add the equal sign and then we write slope, open parenthesis. So it's asking what is our y. Our y is the, or y is our absorbance. And then we put a comma. And it's asking what is our known axis. Our axis is the concentration of fluorescein. And here it's our slope. And it matches with the slope we found here on our equation. Now we ask for the intercept. What did I type? Intercept. Okay. Intercept. And then the first we add our y's. Our y's are the absorbance. I have to always remember myself because this can be quite confusing. And then comma. And then the axis. Our known axis is the concentration of fluorescein. Very well. So here is our intercept. Here's our intercept exactly as we had found it on our occasion. Okay, so very important. We're going to use it pretty soon when we calculate the concentration in our samples. Okay, so group 2 had a few samples. Let's look at their setup. So they had samples collected in location 1 and in their first replicate, right? And they pipetted those 3 times. We call them pseudo replicates or analytical replicates. So we pipette the same sample, whereas the true replicates are those that you collected in the field. You collected 2 samples in the field, or 3, so you have 2 or 3 replicates. Those are real replicates, are your field samples. In the lab, when you pipette 3 times, that means your pseudo replicates. It helps us look how well your pipetting is in case there's any mistakes. It helps illustrate mistakes, but technical mistakes in the lab, but they are not true replicates. So that's why we average them out here when we are doing our calculations. So let's say group 2 had samples called location 1. What else they had? Location 1 and replicate 1. So location. I don't remember if group 2 had like samples from the tree, a distance from the tree, or different species. So I'm just going to call location 1 and 2 replicates. And then they had location 1 and second replicate. And then they had location 1 and a third replicate. Then they had location 2. And I'm not going to type it all again. I'm just going to drag and copy, but then it creates different locations, and then I just click here under copy cells. So it's all copy. And then here I'm going to copy this and this just to make my life a little quicker. Then I'm going to average the absorbance for the pseudo replicates. So I need to keep an eye on their map and on the data obtained by the instrument reader, okay? So their first location 1 and replicate 1 is located in C1, 2, 3, okay? Very well. And then location 1, replicate 2, it's in C4, 5, 6. And then their location 1, replicate 3, it's in 7, 8, 9. Is it? Yes. I really hope this is helpful, guys, because it's like, for me, it's quite, I feel that I'm speaking on the dark because I don't really get any feedback to see if you guys are following or not. We'll see. It's an experiment. This semester is an experiment for all. So let's hang in there. So average location 2 should be in row D. Alright, location 2, replicate 2, it's also in row D. Row D, 4, 5 here. And then location 2, replicate 3. They had 3 field replicates, the real replicates. I think everybody will have the same situation. Okay, so you see we have the average, the instrument reading average, and we still don't know. We have the absorbance, but we don't know in terms of microbial activity. So that's what we're gonna calculate, microgram of fluorescein. So we can plug in this equation with these absorbance numbers. So when we plug in our numbers into this equation, we already have the Y number. We need to find X. We need to find the concentration. So we have Y, but we're gonna find X, okay? So let's start by entering the equal sign. We have Y. Our Y is the absorbance. We're gonna subtract by our intercept, which is here. We're gonna close parentheses, and we're gonna divide by the slope, okay? So this is our concentration of fluorescein for this location 1, replicate 1. We obtain 92 micrograms of fluorescein, which puts the sample close to here, where I'm showing with the mouse. So their sample is close to here. Let's calculate for the next one. Again, equal sign, let's plug in into this equation, open parenthesis, we have the absorbance, we subtract the intercept, we close parenthesis, we divide by this slope. Okay, 76, let's do it again. Equal sign, open parenthesis, select the absorbance, subtract the intercept, close parenthesis, divide by this slope. We're just replicating this equation here that lets us find the concentration of our analyte or of our fluorescein, which indicates the microbial activity. Let's do it again. Equal sign, open parenthesis. Absorbance is subtracted by the intercept and is divided by this slope. 2 more times. And I'm doing it step by step because then everybody can follow. Very well. So now we calculated the fluorescein concentration of each of their samples. But they had the field replicates, which we can average, right? Let's move this graph over here. We don't need to look at this anymore. All right, so now we're just gonna work with this table here. Let's make it colorful. All right, so now we're gonna do exactly as we did for the soil respiration. I could end the video here because you could just look into the soil respiration Excel processing, but I'm gonna continue. So I'm just gonna repeat what we did for soil respiration. So first I'm gonna create a new table, which I just showed my location 1, location 2. And then it's gonna be the average fluorescein concentration, average fluorescein concentration in micrograms. Okay, so here we're gonna average out the 3 replicates. Here, average for the second location. You see, if you just look into each location, there is quite an amount of variability. So you have samples ranging around 75 and a sample that is 92 within the same location. For location 2, things vary even more. We have a large variation. If we just average this value, we have the mean, but the samples actually have a wide range of dispersion. So we're going to see that on the error bar, okay? So let's calculate our standard deviation. ST dev equals ST dev, and then let's select all the data, and then again very well. So here we have a very reasonable standard deviation, but a much larger standard deviation here because the samples vary quite a bit, vary from 33 all the way to 86. Remember I was talking about the height, like people's height varies so much. So the standard deviation for people's height, it's actually quite big unless it's a homogeneous population where people are all the same height, right? So now what we're gonna do, we're gonna create the bar graph, correct? So let's select here just the average and the locations, go under insert, bar graph, and we already have the label for location 1, location 2. It looks like location 1 has a much higher activity concentration than location 2, correct? Is this true or not? Can we say it's significantly larger, the microbial activity in location 1 versus location 2? I don't know. It looks like at the first look, yes, but because there is a wide range of microbial activity in location 2, it's possible that they are not significantly different. All right, so let's add the standard error here. So we go under add chart element, error bars, more error bars options, custom, specify values. Let's do the upper value first and the lower value first. It's the same value, but the errors, the error bars go positive and negative just to show the dispersion distribution. All right, so here we see that the dispersion of data across location 2 is quite large, overlapping with the dispersion of data in location 1. So technically, we cannot say that location 1 has a much larger microbial activity than in location 2. So we have to say that they have similar microbial activities. So I want you to put the title on this graph, okay, and with the unit that we're measuring, and then we will, you're ready to put on your poster, okay? Here we're showing data as concentration of fluorescein. Some people like to show it per gram of soil, per kilogram of soil. So all you had to do is to divide by the fifth, I don't remember how many gram, 1 gram. We used, I don't remember if it was 1 gram or 2 grams. So you can divide by the amount of grams you used on this exercise, on this lab experiment, so you express per gram of soil. Whichever way you choose to do, it will be fine. I think this way is fine. If we just show the microgram of fluorescein concentration, which was released during the enzymatic activities in the soil, and we go from there. Okay, so put this graph on your lab report, okay, along with the soil respiration. So we're done in terms of Excel. calculations. It's simple. I may have overcomplicated things. I'm sorry. It's the first time I do this, and never got any training to teach this number and Excel calculations online. I'm improving as we go. Okay, so let me know if you have any questions. Thank you.